That was fun anyways. Thank you for picking it up. Goodbye, cool
So
rock.
not too much dropped off, but I feel more comfortable making red 5 the bio compartment.
Okay.
Roger that.
Okay, ready?
That's a cool whirlwind.
That's a very nice sand vortex.
All right, let's go.
All right.
2 rocks.
Bridge nav.
We won't stop him now.
Two 0-meter step, bearing 017.
Take pictures of the pebbles. Could you take pictures of the pebbles?
Mark Legg writes that this is a very significant location on the tectonic boundary between the inner borderland rifted area and the outer borderland former subduction forearc, and that we may find some very interesting rocks from deep in the crust.
Ooh.
That's what I like to hear.
Turning, jogging down. Assorted pebbles and cobbles. Evenly spaced on the sediment here.
We passed by a rock there, it looks like. Big rock.
Yep.
On the right-hand side?
Over
Oh, yeah.
on the side.
In the Argus view.
Looks like there's more coming up ahead, though.
We hope
It should
so.
be. We're heading into some pretty steep terrain.
Are these stocky things on the floor, are those sponges?
Video zoom.
Could, maybe we could zoom in on the white sticks. Pieces of bamboo coral, or no? I don't know what those are.
Bridge nav, another step, same bearing.
Don't think it's bone.
Another step, same bearing, please.
Wood? No. It would
Yep,
be
got it.
twigs.
Come wide. Video zoom.
Does that look like bone?
It
I think
does.
it's antler horn.
The shape doesn't really look right for bone.
Yeah, what would that be? That doesn't look too ribby.
Pieces of wood?
Yeah, I would guess that.
Well, that
A
might
lot
be
of it.
interesting to collect. Some wood.
So
Yeah.
you're interested
I
in a
bet
sample?
Greg would like the look.
Bridge nav, hold position. Okay. Come wide. Thank you.
Greg, you're there. You think
Thank
we
you.
should take the time to
There's
collect
one that's
some
round over there, like a disc, right? Go over that away.
collect some of these? Yes, he's watching. Do you think we should collect some of these white objects? Please zoom in.
The bigger
Let's zoom in
disc
again.
there.
I'm working on it. Sorry.
A thorny head?
Yep.
Well, the disc would be different, probably.
Video zoom.
Does that look like bone? No, like a vertebral disc or something like that.
Not really.
Well,
Look
why
at
don't
the
we?
stick? Look at the stick thing next to it.
Could this be bone?
I think they could be bone. They don't really look like wood.
There's another
Exactly.
piece to the left. I'm gonna try to. Oh,
Oh,
that guy's in the way. Sorry.
Oh,
It's
Is
like
that
a rope.
well, right behind that, there's something interesting.
oh, I don't even know. We're not in the sanctuary, but what's the protocol if we think we're collecting bone?
I honestly have no idea. Do you want me to try and find Dwight before we
Or
sample
or
it?
or should we collect it?
Greg is saying it looks like bone.
But no obvious creatures.
What is that? Like
Yeah, though we're not
3
seeing anything
centimeters?
colonizing it, but
I think it's an issue if it's like a whale bone, like a marine mammal, but we don't really know. Would you like me to try and find an expedition leader to get some guidance?
yeah, I'm sort of tempted to move on unless Greg's feeling like he really wants a piece.
It's up there behind that other rock.
So Greg, yay or nay? Should we keep going? Who wants to look at the ones to the left?
Video zoom.
If we don't see any obvious organisms on them, that they're less interesting to us. So,
These guys.
although, here, let me just like these here.
Far
Oh,
left,
okay.
Kylie. Yeah, that curvy one. Try to get you in. That's full left for me. Do you have more zoom, Al?
Is there any more
No.
zoom in there?
Well, I was going to say I can try to get that guy in a little bit better.
Yeah, I do, but not a lot more.
Oops. Okay, sorry.
You could pick a piece up and
Look
look
at
at it
it.
really close.
Yeah, I'm not really seeing much of anything. No, not even maybe a hole or two, but we're going to pass.
Okay.
If Greg's not too keen on them, I think we should just keep going.
Okay. Roger that.
We can make a note that we saw some bone.
Might have been Jimmy Hoffa.
I'm going to adjust bearing to 028 for the next step. 028. You ready? Bob's not giving up the arm. He's just standing by just in case. Yeah. Really? You sure?
No,
You sure?
I'm ready. I'm ready.
All right. Bridge nav. Get a 20 meter step bearing 028.
Yeah, so we had passed over more of that, like,
Yeah, it's
Yeah.
been scattered for a while.
yeah.
Whatever it is.
What does this rock look like, Kira?
Sedimentary, maybe some volcanic. Doesn't look like phosphorite.
Nautilus Live. This is Peaker Histu, copy.
Oh, rope,
I can
It's
hear
rope.
you.
Hey,
That is
Pete,
rope.
good morning.
rope.
What's wrong?
Good morning,
rope
Lisa.
right
Is
there.
that Lisa?
Okay.
This is Lisa.
Look out in front.
Hello.
Look out in front
Okay,
of us.
going. Yeah.
Pan
Morning.
up, pan up.
Okay, Bob. Do you want me to hold?
Keep panning up. Keep panning up. Keep
Oh.
panning up. Keep panning up. There's
Look at that.
A lot of
Thorny heads.
and
Different
crab.
crabs. I think Mary Wickstein's on. She could tell us what crabs we're looking at.
I just want to keep track of that line in case it comes off the bottom.
Oh yeah, see the fishing line on the right.
Okay, so just stay to the left of it.
We want to keep it in view.
Keep my
I don't want
heading
to.
towards
I don't
it.
want to. Yeah, I don't want to lose track of it in case it starts to come off the bottom.
Okay.
Is that a sea pen there too? Polypterus maybe?
So plan is
You
look
wanna
at
zoom
it.
Up, look, pan, and look at that thing.
Okay. Zoom in now.
Or is that as best diploma?
We'll have to zoom in.
Yes,
Might be his best diploma.
down.
Right, okay, sorry.
Yep. We're so much shallower. Wouldn't surprise me if it's other things too. But the best diploma lives at this depth, so it could be that, which is a carnivorous sponge. The rocks have brittle stars.
Can we get more zoom on that?
On the rock itself, or is that okay?
That's good.
Kind of see the whole thing.
Looks like it could be the sponge. Maybe Steve Moskovitz can weigh in.
Do we want to hold position? Because we're definitely moving. All right, bridge nav, hold position. Yeah.
Lots of life on the rocks.
Are we still IDing the sea pen? Is that what we're doing?
Do we want to sample that?
I don't want Argus to be out in front either. You just need to communicate more what we're doing.
I don't know. This doesn't look
So
like
I can
a
back the
pretty
ship
good
up if you
sample
like.
for chemistry.
No, I think it's okay.
Okay.
If there's line, we don't want.
Yeah, for biology, it would be pretty interesting to study like the
deep unknown.
deceased or dead portion and
Okay.
compare it to the live portion.
I gotta get moving.
But I think we could keep going.
Yeah, zoom out.
Okay.
If we're gonna, if we're stopped, are we stopped?
Right now. The ship is stopped. Argus is swinging. We're just keeping an eye on this line.
Is it? You may not want to get this close to the line to sample that sponge.
Science chat is saying it's his best
The
diploma.
problem is if the line comes off the bottom. I don't care if it's on the bottom.
So what are you looking at, Lisa, that you're interested in?
The long stalk.
Right there, and right centered in our view, pretty much.
Is an Abestipluma sponge. Predatory sponge.
You want to sample that?
We're gonna have to move up on it. We're pretty far away.
Okay. Do you want to step Argus back, or nah?
Well, we can. We're still in the picture there, so.
Video zoom. I mean, sorry, unzoom.
Unzoom.
The other zoom.
And then tilt down so you can see your basket some, and then move up on it. But don't land on the line. Just land off to the side.
So, Pete, if you're there, if you want to talk to us while they're adjusting to take a sample, that would be okay. Are you there? Maybe not.
He is dialed in, has been for just over 6 minutes. Pete, if you hear us, please talk to us.
Sorry.
Get off of that rock. Go that way. That's good. Plant there.
Nautilus, this is Pete. Is this a good time to ask a few questions?
Sure. We're just settling in here to take a sample, so.
Don't get too close to it because I got
It's a great time.
The big praying mantis grip there. It's hard to work and too tight.
Okay, it's just not really sitting.
Yep. Back up if anything, because you like.
Pete, I have a feeling you don't hear us.
That's good.
Oh, you're right. Maybe he doesn't hear us.
So, what's the plan here?
We'd like this sponge right there.
And how are we going to get it?
We are standing by for your questions.
Are we going to put this in red 5, forward bio box?
Yeah,
Video's in?
red star, the forward red 5 box.
Yeah.
And you want the whole thing, or
Yeah. Yes, please.
including the base, right?
If you can.
Well, so now the coral cutter makes it more difficult because it's going to want to snip it and not grab the whole thing. But we can see if we can.
The bottom part looks different. I don't know that it's dead, but it's got a whole different collection of associated organisms on it. If it gets cut, that's okay too.
Okay. Well, it's going to.
So I don't think Pete's able to hear us. I don't know which line he's listening on or if we're not transmitting
Yeah, I think
properly.
audio's on it.
He should join the science chat, I think.
Okay, zoom out.
Hi, Lisa. This is Pete. I just had a temporary disconnection. I don't know what happened, but I do hear you now. Thank you.
Okay, that's good. You have a question?
Did we get a good picture of this? Are we good?
1 second. All right, good captures, yes.
Okay. Racking the camera
Zoom
in.
out.
Into forward bio box,
Camera
red
in.
5 if you can.
Okay.
Sure. Okay. All right. Tilt down. Tilt tray out.
What's the number?
This is NA124-199.
Hey, Pete, I see you're still dialed in, but we do not hear your questions.
Yeah, thank you. I'm standing by and waiting till everybody is secure. I know how complicated it can be out there.
Well, this is the time. This is actually a better time to ask while we were doing the sampling.
Well, then let's ask away.
Yep.
Well, thank you so much for letting us join you. I have Elijah here as well as Sienna. And Elijah, you're calling in from Singapore, right? And I believe it's about 1:30 in the morning. So they're really, you have a captive audience. And we have a few questions actually that they wanted to run by you. One thing is Elijah was wondering how much practice does it take to get dexterous with the Herc arm?
Oh. Well, that's a front row question.
I think you can get somewhat proficient in a pretty quick amount of time. But to get like really good at it takes years. You can get the job done in a couple of weeks of practice. Right, Kylie?
It's like
Yeah, actually,
full focus on the books.
While we're talking about this, so just as to share with the students who are online here, there's the so-called front row, and those are the folks who are actually running the vehicles. And if it's not too much trouble, I would love to hear your names. And then if you want to just say 1 or 2 short sentences about how you got into this, that'd be great. Thank you.
Sure. Yeah.
Go ahead, Kylie.
I'll start while I'm stopped here. I'm stopping here. Sorry, that's the camera.
So, Peter, most of your questions going to be for the technical people up front. I'm just because they're the ones who actually have to move us now.
Let's get a move going, and then we'll
this
answer
is-
that question. How's that? Okay.
That
Kylie?
sounds great.
Okay.
We can go to the back row
Sure.
and hear
Cool.
from the scientists, please.
Thank you.
Sounds good. Thank you.
You go first, Lisa.
Oh, how we got into this science?
Who you are.
Yeah,
Ooh,
Lisa.
something about you.
Well, my interest in marine science came as a high school student when I attended a program at Humboldt State College that NSF had for a 16-year-old.
We not no sideways action.
So
I want to know what
that
this
was marine
line's doing.
science. And then my love for the deep sea really started with Bob Hessler's deep sea biology class when I was a graduate student at Scripps Institution of Oceanography. So having really good, and Gary Breska was the Humboldt State instructor. So having some really inspirational instructors made a big difference for me.
For everyone at home watching on channel 3, the bottom left of the quad, I have the pilots and the navigator in the front row. You can see Bob moving his hand around, but the scientists are in the back row. And that'd be Lisa in the middle, and Devin waving just an arm. We only have Devin's arm.
Kira, you want to say how you got into this?
Sure. My name is Kira Meisel. I actually work at the U.S. Geological Survey as a marine geologist studying marine mineral deposits. And when I was young, I wanted to be a doctor. I thought that that was the best way to use science in a career. And then during my undergraduate, I ended up doing a research thesis in environmental science and realized that I could use science to study the earth and earth processes.
So it's like a long line or something.
And then I eventually found my way to marine
Okay,
geology.
we can zoom
and
back out.
really liked that there were a lot of remaining questions about deep sea minerals. So I tried to keep my mind open.
Can we turn a bit and kind of look up, see what's going on there?
And now I'm lucky enough to be here. How about you guys?
So my name
What's
is
happening?
Gabriel Castro.
Can we zoom in on that, Al?
Yeah,
Just see like there's something go up a little bit.
At the university when I started doing research and just loved every aspect of it, the discovery portion. And in chemistry, I fell in love with it.
On there.
And so that took me to graduate school, thankfully to Scripps, where that exposed me to this community.
All right, so they both go up that direction.
Great scientists, and
I don't know what that is. Right
in
there.
the laboratory of Paul Jensen now as a postdoc, because of his involvement in this project, I am here today, and this is my first cruise oceanographic expedition, so I'm really happy
turn
for
that
that.
way.
How about you, Devin?
Okay, which way are we going?
Yeah, so I'm Devin, and
Devin, if you can push over to your left, they'll see you. I'm sorry, I can't pan the camera that far.
oh,
I might be able to give you another camera.
What's that?
all right,
I'm talking to the back row.
Okay, so
Here you are.
I'm Devin, and I've had a general love for all things outdoors, but particularly the ocean. And during my undergraduate education, I actually did a field studies trip to Australia that took me to the Great Barrier Reef, and just diving along there, snorkeling, and seeing all the organisms in the ocean there had really got me into it. So then when I came back to undergrad, I started working in a marine laboratory there and had a love for conservation, and that brought me to Scripps working with Lisa.
So we have a really cool dive track that we have made a little bit of progress on, but I think it would be nice to maybe do a transect or start to see some of the more of the geology along this track.
Right now we're hung up by all the fishing line and trying to figure out whether any of it leaves the bottom and goes up into the water. It's quite dangerous for the ROV to get tangled up in this. And this is one of the hazards of shallow diving. We haven't seen much fishing line because all of our dives have been below 500 meters. We've seen a little, but this is a lot more.
Are we near a marine protected area?
We are just at the edge of the sanctuary. There tends to be a kind of a aggregation of fissures just outside of marine protected areas, and this may be the product as all this line cut up on the rocks.
Wow, that is really wild. We Yeah, thank you all for sharing that. That's fantastic. And we'll leave the front row alone for now. But I have a couple more questions if you all don't mind. Sienna asks, what outcome would be ideal from today's exploration?
Well,
question.
that's great. Yeah, we would love to find phosphorites here in various forms, whether they're nodules, rocks, pebbles, whatever. And we'd love to find rocks that are covered with life so that we can collect those and quantify those. And I know that Gorgonian corals are a big target for today. That's one of the reasons we're diving so shallow. We haven't seen too many Gorgonians yet this cruise. And the Jensen lab is after those to look at the associated microbes and their biopharmaceutical potential. Kira, you want to say what your aspirations are for the day?
You covered it all, I think. Did a good job of summarizing everybody's goals. Yeah, certainly am excited to see the form in which the phosphorites in this region take. I know we have several other geologists in the science chat today who would love to observe the different formations here along this important tectonically active region. So we've got a lot of stuff to see along this cool dive track, and yeah, I think you've done a good job summarizing.
The primary purpose of this expedition is to characterize the animal and microbial communities associated with the mineral-rich substrates, phosphorites, and iron manganese crests. So different depths and exposures have different precipitates. And so where we are today, we're hoping to find phosphorites.
This is very cool.
Oh, that's very cool. Thanks. We got another question, though, that was asked. Why aren't the rockfish darting away from the ROVs? Is there a specific light source that you employ that the fish can't see, or is it just because they naturally don't move too much?
Probably the latter. Rockfish in general, these are probably thorny heads, not rockfish. It's hard to say, but rockfish generally like higher oxygen levels, and we're already at 15 micromolar down here, which isn't too much. So the thorny heads have a lifestyle that tends to conserve energy. Not sure what they're seeing in the way of light and whether they're sensitive to our lights or not. They certainly have big eyes, so they should be. Not too sure what they see. We have a vase glass sponge, a whole series of crabs here. These look, they're lithodids. We got 3 walking legs on these rather than 4. You guys see the crabs?
3.
Are we still hunting for fishing line?
Yeah, I think that was a Strongylocentris fragilis pink sea urchin in the back right there. Ooh, nice jelly.
Like this depth.
Okay, we have a step in.
But not a lot of movements, pretty characteristic of deep sea organisms, right, Lisa? Low and
most of
slow.
them don't move fast, although some surprise you. You can see a brittle star sitting in place.
Yeah, we've seen those brittle stars booking it.
Usually they're sitting and don't move, and then all of a sudden they're off running.
All right. Science, there was a lot of line, so we were getting a feel for where it was. We think we're past it, but we're cautiously moving forward now.
All right, thanks, Aaron.
Your yellow sponge.
Yeah, Staurocalyptus.
With a
Staurocalyptus.
And then, yeah, the one on the top.
Is that like the one we collected last time, kind of?
We did.
Yeah, we did. We have collected, I think, both of those.
There's some solitary corals, some cup corals there. seen too many of those. Desmophyllum dianthus. It's the cup coral.
Kylie, we're
Interestingly enough, I think Desmophyllum and Lophelia are the same thing.
Very pretty.
Did you get a picture?
Oh, yeah.
Bridge
Yeah,
map.
this is gorgeous.
Another 10 meter step.
Folks, I think we'll hang out with you for about another 7 minutes.
You have more questions?
Yeah, if you don't mind. Isabel Baker, who's actually a graduate student here at Harvard, is wanting to know, Lisa and you all, which do you think has been better explored by ROV, the western coast of the United States or the eastern coast?
Oh, wow. Well, different ships spend more time on different coasts. The East Coast has a much broader shelf and slope, and the West Coast is much more abrupt and drops off quickly because of the subduction zones and tectonics. So, you know, the East Coast is bigger if you're thinking continental slope. Honestly,
Yeah, I think
I would say they're
cool.
both relatively surprisingly poorly explored, given that they're part of the United States.
Yeah.
But maybe
siphonophores, right?
kind of equal. I don't know. What do you think?
Zoom in.
Yeah, I agree. I think it's.
What do you think, Pete? There's a Dromelia, colonial siphonophore.
No, you know,
benthic.
This really is cool looking, by the way. What a neat
site.
In?
Did we zoom in on the Dromelia?
Wow.
So this is attached to the bottom, but they can swim. I've seen them move around. Colonial siphonophore, I believe.
I love them. Hands down, they're some of my favorites. So
Kinda
cool.
Spooky.
I know, right? Do y'all see that? They're just gorgeous. So neat.
Remind me never to hire you as a camera op. Joe, Bob.
See all the
Wow.
Oh, Alan. Alan.
Pretty cool.
That's beautiful. You know, Lisa, I think that I wonder if there's been more ROV exploration on the West Coast, but maybe equal or slightly more sort of typical oceanographic work on some parts of the East Coast. But I would bet you're right. Both are woefully underexplored.
Yeah, and it's also a matter of whether you're talking about the bottom or the water column. You know, there's some really important water column survey programs. Coffee, for example, on the West Coast. But the Atlantic in general is far better studied than the Pacific.
Like
the much longer oceanographic.
Like the ones we've seen before are going to town like that. But this one, you would think it would be a regular movement, but it's
Greg said the polyps at the top are the propulsion for this organism, and so once they do detach, it probably moves them around. You can see them pumping.
That is very, very cool.
You know, this is Sienna asks a question for you all. Can you describe what happens if you find something really interesting that is just not related to the specific purpose of your exploration?
You mean like a fossil whale? What happens?
For example,
what happens is that everybody stops and spends some time with that amazing phenomenon, whatever it may be. A few dives ago, we ran across what looked like a fossilized marine mammal in some carbonate, took some samples, and did some flyovers and video. So I think this is an exploration cruise, and we're open to looking at anything interesting along with the primary mission.
Yeah, but it is an important question because there's a lot of objectives on a single cruise, and so you do have to balance your time, and it's always a interesting challenge to try and meet your objectives and explore. But that's the fun part about it, is getting to do a little bit of everything.
Yeah, absolutely. Absolutely. And I think, you know, if I may say so, you know, it's each of us has our own particular expertise or point of view or skills or history or what have you. And I love being at sea with people who come from all different walks of life and bring different things to bear. And that's true for the engineers and camera operators and stewards, and just everybody has something to contribute.
It's kelp.
Or line.
I think it's kelp, right?
Kelp?
Is it? Zoom in.
It does not look like kelp.
Line. Yeah, this place is covered in this stuff.
How do you spell the Dromelia again, Lisa?
DROMLIA. Yeah, this cruise in particular is a really good example of that. We have people, microbiologists, and we have geologists, and we have invertebrate phylogenetic specialists and ecologists. So we are kind of a mishmash of interests, but they all come together very nicely and allow us to, on any one feature, like one rock, we can look at it from many, many different perspectives. And then we have a great technical support here.
Oh, absolutely, right? It's amazing.
Our mappers have been phenomenal, really helping us find all these great spots to go to.
You know, on any expedition like this, Lisa, whenever you and I go out or we go out with our colleagues, if things are going well and it looks easy, there's usually an awful lot of hard work behind it.
Yeah, don't do it.
Paragorgia?
Is that Paragorgia?
I'm going to zoom out a little bit, Bob.
Okay, Lisa, before we let you all go, if I could ask you one question. There's Isabel would like to know, what's ship life like in the time of COVID? What's different this time from your normal expeditions?
Can you see us?
They will in 1 second.
We see the seafloor, and we did see you in masks.
We're all wearing masks all the time.
And
all the time.
Fewer people on board.
And there's fewer people on board.
Channel.
So we've had to have reduced operations. So this is why we're only diving 8 hours.
And everyone on the ship had to go through a 2-week quarantine prior to coming aboard.
Right. We all quarantined, so now we're a ship bubble.
Some of us have been on here for a long, long time without getting off.
Sorry, no port. When we come into port, we just stay on the ship.
Oof.
Longer rotations
brutal.
too, so that they we didn't have to have too many people having to go into quarantine.
Making a dog leg there.
But I think we all agree it's worth the sacrifice to be able to be out here.
Oh yeah, I'd do it again.
Very special experience.
I'm thrilled to be here. Happy that we were able to come out despite all the challenges.
And really, OET gets a lot of credit for figuring out how to make this all happen, because it's not easy in this time.
And they're doing a good job of keeping us safe.
Yep.
Absolutely. I mean, I'm rather envious of you all, so glad you're having a good time. We're going to let you be now. Thank you so much, all of you, for allowing us to hop in. And smooth sailing and good hunting.
Thank you. Thanks for coming.
Bye.
Bye, Pete.
So we can see, you know, in these shallower depths, we got lots more glass bottles, lots more baby bones, lots of fishing line. It's a different place than where we've been the rest of the cruise.
Yeah. Okay, zoom out.
It's Bob's Nightmare Land.
Okay, well, I think we're going past it.
Okay, we're still in a move, so we'll just keep it slow and steady.
Yeah.
I mean, I think it's probably not good to try to do a transect, right? Because you need to be able to maneuver up and down.
I'm not a fan of high-speed transect with line all over the place.
That's what I figured.
We haven't seen any that's like we were getting like on our earlier legs when we had big 3/4 inch line and traps attached to it. But this is still
scary.
Yeah, well, it can get in the thrusters and then seize up a thruster. Well, that's kind of a problem.
These more sea pens we're seeing on the rocks here?
Have to look closer.
Zoom in out.
Right above the lasers?
They may be.
Looks like the same as
Vestopluma again?
Or a Vestopluma.
Yeah.
Yeah, it's interesting, these
Zoom in.
large boulder formations. It could be sedimentary rock or...
That's Vestopluma.
Yep.
Yesterday we cut some mudstones from rock that looks similar to this. So it could be...
There's a skate right back there, it looks like. Or is that a
It's
rock?
just a rock.
Oh, never mind.
There's a lot of rocks around here that want to be fish.
Yeah.
Any of these flat slabby rocks look promising?
Not really. I'm not seeing that texture we were seeing several days ago.
Is it 3.8?
Yeah, keep it 038. So science, is the intention with BB and then to BC, is the intention to come up on a possibly flatter area, or do you want to keep kind of creeping along contour to keep...
Well, there was one, could you put the backscatter on, please? There was one more sedimented spot that we had thought about for coring. But it's way up there, yeah. So the waypoints are intended to keep us along the steeper part of the route, and maybe in a few cases up on rims where we might see more biology.
Pile of anemones.
Let's go see if this is a little kelp fall or a dead animal or somebody. Usually when we get all the pink urchins piled
that looks
up,
like kelp.
there's some kelp.
Yeah.
Let's take it. Can we zoom in on that? Just get some good photographs.
Hey, zoom in now.
Maybe a slurp.
Strongylocentris fragilis.
Yes.
It's a nice word.
They love to eat kelp, and they seem to get it at all depths here. Zoom in and see how. The last kelp fall, we had this amazing ball of Dorvalaid polychaetes swarming, thousands of them. I'm not seeing that here. Wait, well.
Are you interested in doing something with this?
I'd like to just look at it a little more closely.
I'll get closer.
So you might have to inch up there.
Get the porch light on?
If it's not highly decayed with lots of other things around, we probably don't want to slurp it. But if it looks like there's a lot of different organisms associated, we might.
I think I'm going to need it on, though, for these close shots.
Well, that's why I was just wondering if it was on.
Yeah.
Okay, you got more zoomage?
We need. Someday we'll have great lights.
So they're on
look at all those little
there's
critters.
a lot of little amphipods.
Oh, man, a whole bunch of them.
Let's do a
It's
slurp
like ants.
Is that a fish in the top right too? Do you see that eye?
Yeah.
Between the sea urchins.
Yeah.
We copy. We want to slurp there.
Yeah.
Let's do a
Yeah.
section.
Look at all these things on the.
Oh, it's a sculpin.
What are they? They look like gammarid amphipods, maybe.
That looks like a great hiding place. Just like, I dare you to come get me.
Well, hiding under urchins is a thing. Lots of animals do it, actually. In shallower water, their abalone and little baby urchins hide under the big red urchins that might be 10, 20 centimeters across. So it's a Isolinus sculpin behind the urchins, we're told.
What jar would
That
this be
little
going
fish,
into?
little sculpin. Got lots of people watching today.
Can you zoom out, Al?
Thank you.
Okay, let's choose maybe number 2 or 3, one of the fine ones. Section 2 or 3.
This is our 200th sample.
Wow.
Okay, zoom in.
Sample jar is not rotating.
So we don't really need sea urchins up the suction because they will clog it and get stuck. And we know what they are. They are one of the most common animals on the California slope with a distribution between,
I think that'll do.
Sample jar 2.
1,200, 1,100.
Ready for suction?
Shallow as 100.
Don't give it to beans, though. We need more zoom, I guess.
You've got 20%.
We just don't want to. We don't want an urchin going.
Maybe you could nudge. Are you able to nudge the urchins over? And
yeah, you want to kill the juice?
Yep, no juice.
We couldn't get some suction on an urchin, and because they're not that. I mean, they're big enough where they won't go down the tube, but we can pick them up.
Which canister are we on?
2.
Just give it a little bit of juice just to pick it up.
Okay.
Are we on canister 1 or 2?
It is
2.
canister 2.
2.
Thank you.
That's 10%. Would you like more?
Yeah, give it more beans. That's not going to go in the tube. It'll just
That's
hold on
20.
to it.
That's 30.
There's that sculpin getting out of the way.
That's 40.
Roughly high calorie dive, always beans and juice.
I think he's
50.
hanging on for dear life there.
60.
Okay.
A little more.
Yeah, give it. I guess you got to give it juice.
That's 70.
Okay, let off the beans.
Sure.
Okay, zoom
That's
in.
fine. If you just nudge in there, where all those amphipods are.
Ready?
Yeah, can you give me more zoom, Mel? Okay.
Okay, suction's coming on.
I don't want too much of the seaweed.
The rock surface, maybe.
That's 70%.
I guess that's a rock. Maybe it's a... Oh, fish is hiding. Run away, little fish.
I'm not sure.
And a little more of the kelp, maybe.
Is our sample number 200?
Yes, 200.
Thanks.
Then which canister are we going into?
This is slurp number 2.
That's correct.
Let it off.
Sure. Off.
You can
Back on.
You can come down to the right. Sorry,
Okay.
the other side of where this kelp debris is over here. That'd be good.
Okay.
Yeah. Let's get the things that are living on and chewing on the kelp.
They're like underwater ants.
Insects of the sea.
What are they called?
Amphipods. Okay. That's probably good enough.
Okay.
Yeah.
All set.
Thank you.
Killing section.
Okay,
All right.
Continue on, 038. I see your position.
Let's see. We can pretty much go
Due
north at this point, or 009010.
changing heading to 010.
Yeah.
Roger.
Bridge nav.
Gabe, could you get the depth of that previous target?
Can we get a 10 meter step bearing 010? Yep. Did you need the target backup?
Yeah, I was just curious what depth you reported.
You zoom in now.
405, thank you.
Yep.
It's the long arms.
Long arms.
Okay, zoom out.
You want a junk zoom for this, right?
Yeah.
Nope. Yeah, these all look like sedimentary rocks to me, the smaller ones. Which phosphorites are part of the Miocene sedimentary rock in this region?
Zoom in now.
No, this sponge.
Yeah, it's the one we collected yesterday.
Got more zoom?
Was this what the crinoid was on?
Exactly.
Yesterday.
Yeah.
I don't remember the sponge very well.
I don't either.
Yeah, now it's covered with sediment, so it looks different. I think it's from the genus Boesilastra.
Okay, zoom out.
You see any rocks that you think look interesting? Let us know.
Bob, you might be
All we have
left.
so far is that one.
That's true.
Zoom in.
Bridge nav, another step, same bearing.
What is all this layering?
That looks like wood.
Really?
Does.
Another step, 01010 meters.
Maybe sponge. I guess so.
Strange.
Or the little white puff balls.
You gonna zoom in some more?
It doesn't look like carbonate, does it? I mean, carbonate ledges or anything.
I think
like
you
that.
can see the little pores from the sponge.
Maybe. It's woody in texture.
Is that all in?
That's all I have.
I'd get closer, but
Okay.
I think we're good.
all right, zoom out.
Strange.
See, our oxygen is still around a little less than 20. We're just at about
Zoom in
400
again.
meters.
Is that more Besta Pluma? Looks like it.
I'm going to go inside her.
Yeah.
Lots of interesting things on the outside. At first I thought that was
biology.
White mineral.
Okay.
No, it's biology. White.
Yeah.
Sponge.
Zoom out. You zoom in again now? So is all this dead sponge or?
It looks like it to me. It doesn't look like rock.
He's looking at the flat stuff. Yeah, this. That does look like some skeleton. I don't know. Wouldn't be terrestrial, would it? Origin. Cactus.
I don't really know what wood looks like once it's been underwater for a while.
Does this look soft to you, Bob? This thing?
I mean, it sort of looks like wood to me, like those are roots or something. I don't know.
Poke it. We had a suggestion to poke it.
Can we zoom out?
There's another piece of it right there.
Look at that giant. I wonder if that's a mud deposit. I'm not sure. That's really interesting looking.
Do you want a junk zoom?
Yeah, it's not like cardboard thrown off a boat or something like that.
Yeah.
Okay, Alan, you can zoom in.
It's hard. Yeah. Interesting.
Sticking onto that rock next to it.
Stuck to the rock.
Yeah, now it does kind of look like more like a sponge skeleton.
Yeah.
It does. Looks like a pretty old one. A dead glass sponge. There it says. Don't think we want to collect it.
No, it's too big anyway.
Yep. Thank you.
Okay, zoom out.
For people watching, sponges, they're really cool when they're alive, but they have a whole lot more associated organisms when they're dead. And they're a good source of hard substrate habitat. Also usually with lots of nooks and crannies for hiding. So like whales, sponges have a afterlife.
Erin, could we see the overall progress on the dive, please?
Yep, sure.
Thank you. Wow.
That's all right. The second watch always cleans up after us.
Yeah, just not quite seeing the rocked environment that I'm looking for, so I wonder.
Do you still want to try and kind of keep it along here on the way, BB to BC? Okay. Bob, you can go 000.